All right. Does anyone on the internet have trouble hearing, seeing? Okay, we're still experiencing little AV issues with the new system, but they're better this week, and hopefully next week will be better. So this is today code. I have to get used to Maria's graphics. So this Thursday is leap year. Floating meeting controls. It was introduced by Julius Caesar in 40 BC, and the Summer Olympics are held every 4 years on leap years. I didn't know that. That's probably an out of 4 chance thing rather than anything specific. Okay. The other great thing about it is you get a dozen cream donuts if your birth happens to be February 29th. So bring your proof of birth license, and you can get a dozen of those poisonous donuts. Carolina Girl Society, the neuro team crushed the competition. Is that spelled correctly? Crush it with an E? Really? Crushes the competition. Larry, look at that. Again, David, Jack, and the most amazing thing is it's going to be in Durham this year. I don't know if that's the first time ever. That's certainly Joel on the North Carolina Neurology side. So that'll be so we'll have to get our even more out since we're hosting it. Our Michelle for leading the interviews this year. Great work, nominated by Christine. Janelle Montgomery, it's the nod to Michelle, being a really ex-MS patient, make sure she got her med. Katie Moore was on helium answering questions about Huntington's disease. A lot of people in the news. Maggie Soltis was in the New York Times. An article that I read, it's too long to stay in bed. There's no too long to stay in bed. If you can't sleep, you get a chance on the weekend, you just stay there. Katie Peters, an article called Cure on drugs to combat cognitive decline and brain cancer. Andrew Spector was on Health Digest, talking about how beta blockers can affect sleep. Wayne Feng convinced me to start a beta blocker for my blood pressure. So, Andrew, thank you for saving me. Sneha Mantri is a part of a study in movement disorder. Looked at the most bothersome complaint of Parkinson's patients, and it was interestingly cognitive. Brian Rory was principal of R, talking about how to set up a comprehensive eye stroke center. Lots of stuff. Sharon Petrak, Luke Kim Johnson looked at retinal changes in people with mild cognitive impairment. Now, finish with that. Now, here, the did you know? Like, who knew? All you young people hate chocolate chip ice cream, it turns out. And it's absolutely the best favorite. But the New York Times said no one's it anymore. And the best comment was it's similar to cookies and cream, but with no cookies. So it's killing. These are the top 10 favorite flavors. Chocolate chip isn't even on the list, and mint chocolate chip is in no chocolate chip. It's a horrible bastardization of the process. But there you go. It's just terrible. But another article on the Juro spiders time in CNN, and you all know they're coming. If some of you don't know about the Juro spiders, just ask the person next to you. They have this adaptation where they're not afraid of hard, loud noises because of some mutations. So they're going to live right on the streets in your houses. Boom, 2 more years, they're here. Look at how these things are. And they use their nets as a parachute, as a means of, their nets are 10 feet wide. And when the wind comes, it grabs the net and it parachutes them like mile to mile. And they're coming from Georgia, and they're going to be in Durham in about a year, and they're going to be all over the place in about 5 years. You're going to go outside, things are going to be everywhere. They're not venomous, but that hardly matters. It's like some snakes aren't poisonous, but they're still snakes. They grow. This is today's CME code. And today's round speaker is going to be introduced by our own Andrew Spector.
Good morning. I have the pleasure of introducing our speaker this morning. Dr. Aroke is our third in the Duke Neurology Diversity and Inclusion Grand Rounds lecture series for the year, and tenured associate professor at the University of Alabama School of Nursing in Birmingham, where he directs both the PhD and DNP PhD pathways. He started his medical as a nurse, came to Duke, worked in our cardiac ICU, and then did CRNA here. Finished with Master's of Nursing at CRNA Duke in 2011. Then went up to UMass, got a PhD in nursing with a focus on pharmacogenetics. He's the recipient of multiple NIH grants that fund his primary area of research on how epigenetic differences lead to health disparity, particularly related to pain. And he's a guest of Dr. Carleen Moore, our pain researcher. His work led to recognition as of the year by the American Association of Nurse Anesthetists Foundation 2021. And among many honors was the Annie Beery Award, Standing Leadership from the Duke School of Nursing. Very pleased to welcome back to Duke, Dr.
All right, thank you. Let me share my screen. It says screen share is disabled.
Gotcha. Okay, you should be good.
All right, can you see my screen okay?
Yep, looks good.
All right, thank you for the great introduction. So I will be sharing with you some of the work that we are doing looking at the role of epigenetics in what I call social determinants of pain. And my hope is that for today's presentation, by the end, let's get a little understanding of what it means to be, when I say social determinants of pain, define epigenetics, but even more important, start looking at the role of epigenomic changes in pain disparities. This is my disclosure slide. I won't be making any money from this. And I'd like to just acknowledge some of the support I've received for this work. Again, initially, I was funded through the ANA Foundation, and then the National Institute on Minority Health and Health Disparities, and then currently funded through NIAMS. But most important, always say we are always thankful to our research participants and my collaborators. So thinking about today, again, you may be wondering, what the heck do I have talking about pain to neurologists? But if you really look at pain, it's one of the most significant public health problems that we face today, such that, most patients go to seek care because they are in pain. Globally, you are talking about 1 in 5 adults suffer from pain, and the prevalence is only going to continue to increase. Here in the United States, the prevalence is the same, about 20%, and it's estimated that about 50 million people in the United States suffer from what we call high impact pain. Other studies have cited about 100 million people. And of course, that comes with significant cost. But of interest to me is really low back pain. Why? Because if you look at all the pain conditions, arguably, low back pain is one of the most painful and disabling chronic pain conditions out there. And the prevalence of low back pain is so high, such that about 80% of adults will experience low back pain at some point in their lifetime. Chances are that, maybe at the end of a stressful day at work, you're going to have some low back pain. The good news is for most people, it's going to go away. But for some adults, it persists and stays beyond 12 weeks, and we call that chronic low back pain. It's estimated that in 2020, about 619 million people were affected by low back pain globally. And again, the prevalence is only projected to increase. When you looked at years lived with disability, low back pain is the leading cause of years lived with disability. But what really intrigues me the most about low back pain is not the prevalence, it's not if somebody has a herniated disc, but it's this subset of individuals who have what we describe as non-specific chronic low back pain. Non-specific meaning that if you were to do a CT scan or an MRI, you would not see why they have low back pain in the first place. It's not related to tumor, it's not related to trauma, it's not related to any injury. And so why do people develop pain that persists, even though, again, as providers, we cannot tell why they have pain in the first place? And when you're really thinking again about pain, this is how I studied pain when I went to anesthesia school, right? We really focus a lot on the pain pathways and the different receptors and the different pathways going to the brain and back and pain modulation, which was all great. But what this doesn't really capture is the fact that, one, it's such an individual experience, right? Again, when we're thinking of pain as opposed to nociception, it's very, very subjective. And the fact that pain is invisible, right? Unverifiable, and poorly understood means that for us healthcare providers, we have a lot of leeway in writing on that script, whether we want to believe the patient if they have pain or not, but even more importantly, how we project about what we think about the patients. So it gives a lot of room for stereotype, for stigma, especially when you start looking at individuals of lower socioeconomic status and racialized minorities. And talking about racialized minorities, we know pain affects individuals of all population. But across most chronic pain conditions, racialized minorities experience greater pain severity and pain disability compared with non-Hispanic whites. So in this study, we recruited community-dwelling adults, again, with non-specific chronic low back pain. And as you see here, we're basically showing you that, yes, again, as of 2020, non-Hispanic Blacks in the United States still experience greater pain severity and pain interference compared to non-Hispanic whites. So, of course, my first thing was to see, can we explain this through some of the key factors that predict pain, such as depressive symptoms? So we saw that, yes, when you look at both Blacks and whites, there is a positive relationship between pain interference and depressive symptoms, right? That old adage that depression hurts is true. Greater depressive symptoms lead to more pain interference and pain severity. But if you look at the strength of the relationship, it's not the same in both groups. Even more interesting, we wanted to look at what about the role of socioeconomic status. So instead of just looking at income, we asked our participants to rate where do they see themselves relative to everybody in their community. So we call that the subjective social status. So I'm basically asking that on a rank of 0 to 10, where do you see yourself on a social scale relative to everybody? And you see that again, there is a relationship between pain interference and subjective social status, such that for white individuals, as their social status increases, pain interference decreases. But interestingly, for Black individuals, we saw that as social status increased, pain interference only increased. Then we looked at the relationship between social status and depressive symptoms, and it was the same. So basically, with a moderated mediation model, what we were able to come out with was that, well, it does appear as if social status is protective for white individuals, but not for Black individuals, such that for Black individuals, as social status increase, depressive symptoms increase, which also increase pain interference. But for white individuals, as social status increase, depressive symptoms decrease, which also decreases, pain interference. But that's not really the key message I wanted us to focus on to them. That was just to lay the foundation for us to really think about pain disparities. When I went through nursing school, I always said, if you look at most chronic conditions, including chronic pain, if we doubted any risk factors, you could easily assume in your best guess, smoking, alcohol, or being Black. right? That was one of the key things, again, as a nursing student early on, trying to, if you had no answer and there was a chronic condition, I say, what are the risk factors? And we have known these disparities for a long time. And the key question then is, what is the something that is so damaging to the health of underrepresented individuals? What is it that creates these disparities? What are the root causes? When we think of disparities, again, generally, what comes to mind is social determinants of health, which is good. Social determinants of health account for about 70 to 80% of health outcomes. So we basically took this model and said, okay, we know that social context, right, of where people live or have been and is a powerful determinant of pain. Social factors determine whether people will experience pain and what they think, feel, sense during that pain experience, and how they behave and communicate about pain with each other. And so through social determinants of pain, we really wanted to say, let's apply the model of social determinants of health to see if it applies squarely to social determinants of pain. So that is really what we've started doing here. And we basically saw that yes, it's true. Individuals of lower socioeconomic status tend to have greater pain interference and greater pain intensity. Also, lack of social support worsens pain outcomes. Living in a poor neighborhood has been associated with worse pain outcomes, greater pain interference, and pain-related disability. Access to care we know affects pain severity and pain outcomes. And even more important, again, we know that level of education, lower levels of education increases pain risk. This is all good in terms of, again, explaining some of the determinants of pain. But it doesn't really tell us something, because if you look at pain disparities, especially racial pain disparities, and think about the fact that race is a social construct, not a biological variable. I mean, despite our best effort to identify the gene that codes for race, there is no single gene that codes for race. As such, it begs the question then, how do social experiences, how does these social determinants really get under the skin to create these health disparities? You know, how do we go from living in a poor neighborhood to having worse pain if there is no gene linking it? And could epigenetics be that bridge? That is really what I focus my work on, trying to look at epigenetics as the bridge between the environment and the biology. So what is epigenetics? Again, back in the 1940s, this was coined back in 1940s by Worthington. At that time, he was a developmental biologist. At that time, there were two main schools of thought in developmental biology. And the key question of the day then was, how do we go from a stem cell to a cardiac cell that is very, very different from a neuron? And the two prevalent schools of thought at that time was the preformation school, which basically said, with preformation, they said all adult characters were already present in the embryo, and it simply unfolded as the cell divides over time. We also had the epigenesis school of thought, which basically said, now, it's really not that all characters are already present, it's that new tissues were created from successive interactions between the constituents of the embryo. Worthington said, no, it's not one or the other. It's actually a combination of both schools of thought. And he introduced this landscape theory, where he basically said, as you go from the embryonic stem cell and you divide, as you go through successive cell divisions and differentiation, this embryo unfolds based on gene expression, such that as the genes are being expressed in response to different environmental stimuli, that is how you go from, again, an embryonic stem cell all the way down to a committed cell. So through this successive combination, he coined this term epigenetics to say, epigenetics basically refers to changes in gene expression or phenotype which are caused by responses to the environment without changing the underlying DNA. And these changes in gene expression are heritable, such that they can be passed on from one generation to the other. And we have seen this in tumors, in cancers, and now we are trying to apply it in other conditions, including chronic pain. And when you think of epigenetics, there are three main types, right? But you may be wondering, so why focus so much on the epigenome as opposed to the genome? Like I mentioned earlier, we haven't been able to identify a gene that codes for race, for instance, or that codes for somebody of lower socioeconomic status. But when you look at the life cycle of a butterfly, right, it captures well why epigenetics makes so much sense. As the butterfly goes from an egg to a larva to a full butterfly, the genome hasn't changed. What has changed is the epigenome in response to different environmental stimuli, such that when the gene for the development of wings is needed, that gene is expressed. When genes for development of eyes and antennas are needed, it is expressed. And that is really what happens. Again, the underlying genome hasn't changed, but the expression of it has changed. And that is what is epigenetics. So there are three main types of epigenetic changes. And just to, again, take us by a step back, when you look at genomic DNA, right, again, most of the genomic DNA is found in the nucleus of cells, where we see them in the form of chromosomes, which are tightly wrapped around these proteins called histones. And histone modifications is basically changes that we can tack on covalent bonds to these histone tails. And if you make it such that, again, you have these changes such as acetylation, eucobination, methylation, phosphorylation, it basically determines how tightly the DNA is wrapped around those proteins. And that is going to affect if that DNA can be accessed by transcription factors to be expressed. But if you look at the DNA even much further, we also have these regions in DNA called CPG islands. And those CPG islands are basically areas where we have the cytosines linked through a phosphate to a guanine. And if you have so many of them in a CPG island, such as in the promoter region, it becomes a regulatory area. DNA methylation is basically the addition of methyl group to DNA, especially in the context of these CPG islands. If they are present in the promoter region of a gene, it's going to regulate whether that gene can be expressed or not. For the rest of the lecture, we'll focus mostly on DNA methylation, partly because it's one of the most studied epigenetic modification, but also because most of the studies in pain have, especially in animal models, look at DNA methylation. So again, in talking of DNA methylation, it's basically the addition of a methyl group to a cytosine, and that process is catalyzed by enzymes called DNMTs. You have several of them. And the key basically is if you were to take, a gene such as, if this is your target gene, the promoter region of most genes have the CPG islands. And for that gene to be expressed, transcription factors have to bind to the promoter region, and that gene will be expressed. And that is really what happens. If you have a region that is not methylated, transcription factors can easily bind, therefore that gene can easily be expressed. Increased expression will lead to the production of the target proteins, and these proteins could be inflammatory markers, there could be cytokines, there could be nociceptors, there could be endorphins. On the other hand, if you have another target gene whose promoter region is methylated, then transcription factors cannot bind. And if transcription factors cannot bind, we say that target gene is silent. And if it's silent, it means it's not being expressed. So the proteins which were supposed to be produced from there would not be produced. As a result, the end target response that you expected, be it inflammation or reduced inflammation, if you are looking at the pro versus an anti-inflammatory response, would not happen. So to start, initially, one of the very first human studies looking at pain, low back pain, was by this group out in Canada, by Tejeran in 2011. And basically, they looked at 10 adults undergoing spinal surgery for degenerative disc disease and healthy controls. And they found that DNA methylation in the promoter region of the SPARC gene was actually correlated with expression of that gene and also correlated with pain severity, such that increased methylation led to decreased expression of the SPARC gene. And that is pretty much what we are showing here. This was a very good proof of concept to say, in the context of low back pain, yes, epigenetics through DNA methylation can regulate low back pain or correlate at least with low back pain severity. We basically did that too with our sample, where we said, okay, can we first of all show that for individuals with low back pain versus healthy controls, do we see differences at the level of the epigenome? And what this shows you here again is, using reduced representation by sulfide sequencing, we identified over 65,000 differentially methylated loci, which translated to a lot of genes. Then after controlling for multiple testing, we then annotated these loci or differentially methylated loci to differentially expressed genes or methylated genes. Most of those genes enrich pathways which have previously been associated with things like neuroinflammatory processes, chondrocyte differentiation, and bone mineralization. So you may be thinking again, how does this really apply to us better understanding disparities? And this is what we proposed in 2011. We said, you may have two adults whom at birth their epigenome is very similar. As we go through life, basically, we will be exposed to different environmental exposures, right? Be it stress from life, family, work. But for the Black individual, there is also this additional stress, the stress of injustice and racial discrimination, such that by the time they get to adulthood, their epigenome would be vastly different. So even though at birth the epigenomes were very similar, by adulthood as we go through, the epigenomes may be vastly different, predisposing the Black person to chronic pain. Then we set out to say, let's test this hypothesis if this is true. So when we compared adults with chronic low back pain with Blacks with chronic low back pain with whites with chronic low back pain, we saw that yes, the differentially methylated genes enrich pathways which have been associated with pain processes. So pathways such as the dopamine feedback signaling pathway, axon guidance signaling pathway, endocannabinoid neuronal signaling pathway, which again all play an important role in pain and pain modulation. Also within this, you see pathways such as the corticotropin releasing hormone signaling pathway. But we were not quite sure at this point whether which of these play an important role in Blacks versus in whites. So we did a stratified analysis and basically said, okay, let's compare Blacks separately from whites so that we can identify which pathways are playing a stronger role in whites versus in Blacks. And this is what we are showing here. If you look at the blue signals, those are the pathways playing a stronger role in predicting pain severity and interference in Blacks. And you notice that most of the stress-related pathways rise to the top, which is corticotropin releasing signaling. The white adipose tissue browning has also been associated with stress. The opioid signaling pathway, the dopamine signaling pathway, all of those became way more significant when we look at Blacks with chronic low back pain compared with Blacks without chronic low back pain. As you come down from the bottom up, you see the pathways which are more significant when you look at whites, again, in a stratified analysis, such that we basically concluded that when it comes to racial differences, pathways involved in stress and stress regulation play a more significant role or are more strongly correlated with Blacks, while pathways involved in inflammatory processes are more significant in whites. So we took it a step further and said, well, since we do not, there is not, again, in this analysis, race was self-identified. So let's try to capture something that is not just self-identified, is it? What about stigma? Again, we know that internalized stigma is one of the strongest predictors of worse pain outcomes, right? And one of them is really the fact that we are studying non-specific chronic low back pain. So you have this situation where a patient presents, maybe a provider does a CT scan or an MRI, and there is no identifiable cause, yet the patient says they're in so much pain. That leads to these stigmatized comments, right? And I'm sure as providers, we all have heard of this once in a while in our practice, where they say the pain is just in your head, right? You must be crazy. And the patient starts believing some of these things that providers think and say about them. It must be their nerves, right? Several studies have shown that providers tend to attribute pain experience, especially by women, to psychological factors compared to men. Pain experienced by racialized minorities, again, tend to be expressed or described and explained away as just being their nerves or anxiety. When the patient believes those stigmatized comments, that is what we refer to as internalized stigma. And affected individuals begin to accept these stereotypes, and that has been associated with worse pain outcomes. So we decided to say, okay, let's see if when we compare individuals with high levels of stigma, again, internalized chronic pain stigma, with those with low levels of internalized chronic pain stigma, do we see any differences? Yes, we saw that when you compare low versus high levels of stigma, there's a significant difference in terms of pain intensity, pain-related disability, and stress, such that high levels of stigma positively correlate with high levels of stress. Then we took it a step further to look at the role of epigenetics. Do we see differences in pathways which have been associated with stress or pain when we compare individuals with low levels of stigma and individuals with high levels of stigma at the level of the epigenome in terms of differentially methylated genes? And again, you see pathways, the differentially methylated genes enrich pathways which have previously been associated with stress, such as the hippo signaling pathway. Right? The hippo signaling pathway has been associated not just only with oxidative and mechanical stress, but also with stress-related disorders such as PTSD, depression, and now we are linking it also to pain. So basically what we came, the key takeaway from this study was that, yes, internalizing stigma correlates with worse pain. We know that racialized minorities tend to be stigmatized more than non-Hispanic whites. So maybe the worst pain that they tend to experience may be linked to the fact that the experience of persistent pain and stress and discrimination is getting in this, under the skin through stress dysregulation and altered epigenetic expression of pathways which have previously been associated with stress. So at this point, again, typically when I present to clinicians, they always say, so what can we do about it? So what can we do with this information? Yes, we can try to drug the epigenome. And several studies have done that, where if these pathways or if DNA methylation is catalyzed by DNMTs, maybe we can pharmacologically target those enzymes and reverse it. That is one way. And we have a few animal models doing that. And in this animal study, they basically try to look at rats were inflicted with plantar incision and treated with DNMT inhibitors, basically inhibit the DNA methylation process and also histone modification or HDAC inhibitors. And basically what they found was DNMT inhibitors actually do attenuate hyperalgesia and increase the expression of the mu-opioid receptor gene. HDAC inhibitors also alleviate pain sensitization and hyperalgesia. HDAC inhibitors or histone acetyltransferase inhibitors also increase hypersensitivity. So there is some promise in being able to target DNMT inhibitors and HDAC inhibitors as potential pharmacological interventions. But one of the key limitations of this is that HDAC and DNMT inhibitors are not very tissue specific. So if you were to give it systemically, the effect is going to be systemic. But what can we do today to address it while we continue to research? We know that the DNA methylation and epigenetic changes are dynamic and reversible. We know that stress management definitely helps with pain. And the good news is most of the modalities or stress management interventions that we know today have been shown to affect or change gene expression through epigenetic modifications. So meditation, exercise, having a good night's sleep, all of those interventions that we know work today can actually help, not just through epigenetic changes, but also changes in gene expression to help with pain management. I'm just going to share with you a few studies again supporting some of this. 1 week of running has been shown to decrease methylation level of a BDNF gene. Increase in BDNF expression has been associated with neuroplasticity and decrease in pain, especially inflammatory pain. Swimming induces epigenetic changes in the skeletal muscle. And again, it has been associated with decreased pain and an improvement in pain outcomes. Aerobic exercise has been shown to lower oxidative stress through epigenetic changes and decrease inflammatory processes. So again, basically what we are saying is that epigenetic changes can actually be reversed by things that we already know we are supposed to be doing, which most of us do not do in a way. What about yoga and meditation? Again, 12 weeks of yoga has been associated with a reduction in pain severity, interference, and pain sensitivity. And yoga has also been associated with differential expression of genes involved in neuroinflammatory processes. So again, what we are saying is being engaged and involving your patients in some of these non-pharmacological interventions may actually help reverse epigenetic changes associated with increased or worse pain outcomes. Am I saying that we should put all Black patients in interventions and then white patients in different interventions? No. What I'm basically saying is, as we approach patients, let's not just focus only on pharmacological interventions. Some of these non-pharmacological interventions may be much more effective than pharmacological interventions. So again, in thinking in terms of some of the implications, we already know, and these have been documented for years, that racialized minorities experience more severe and disabling chronic pain compared to non-Hispanic whites. We know that epigenetic modifications regulate gene expression in response to environmental stimuli such as chronic stress. And epigenetics is coming out as a new way for us to really understand how the experience of being Black may be leading to worse pain outcomes, such that epigenetically induced alterations in genes which regulate stress and neuroinflammatory processes may be playing an important role in pain disparities. Of course, more studies are needed in this area. But as we think about, again, addressing pain disparities, I always say we must go beyond social determinants of pain. And this is really why we focus a lot of our work on really understanding the root causes and the mechanism. Because for us to reverse epigenetic changes and promote pain equity or health equity, we have to address the root causes. Yes, social determinants of health are very important, but we must recognize that social determinants of health sit on top of about 100 years of Jim Crow laws, segregation, redlining, and targeting of Black individuals. Again, it sits on top of over 200 years of slavery and post-emancipation structural racism. So while we continue to investigate the mechanisms by which these experiences get under the skin, it's also very, very important that we have interventions to target some of these structural changes, some of these structural and systemic problems which undermine or underline the social determinants of pain or social determinants of health. So how can we advance pain equity? One, we have to go past race. which is really what most of my work now is focused on. We've seen that the problem is not the color of somebody's skin or the texture of their hair or the shape of their nose. But instead of focusing so much on race, we have to address the concept that race represents, which is the effect of things like racism, injustice, and discrimination. Lack of diversity and representation in the genomic studies continues to be a big problem, especially when you look at things like the reference genome against which everything else is compared. Right? So again, increasing diversity not only in research participants, but also among investigators is very important. Nearly all studies involving epigenetics, genetics, or genomics means that you're going to have access to a lot of information about the patient. So that brings ethical, legal, and social implications. Again, as we move forward with precision health, I always say we have to be more mindful. If not, the disparities that we saw yesterday will only persist. Precision health is definitely going to help. Being able to capture the individual's life in a holistic approach is the only way we'll be able to truly target pain disparities, such that we recognize that, yes, some individuals may have greater stress. Maybe it's not just giving them an opioid to treat the pain, but rather prescribing a stress management intervention, maybe what the patient really needs. Integrating lifestyle, environment, and genetic information can foster pain equity, and basically making sure that, again, they have access to care. All the social determinants are good, but for some, we see that epigenetics, again, how can be passed on from one generation to the other. So can we then start developing interventions to reverse some of those epigenetic changes? So these are some references that I included. Most of the slides also have references. I think again, this is my contact. I think we have time for questions.
Thank you so much. Oh, we are going a lot, aren't we? Okay. I'm looking in the chat. Anyone in the room want to lead off with a question? I'll look and see if we've got any questions in the chat yet. I noticed, oh, sorry, Felix, go ahead. Okay. All right. Try again.
I just wanted to know.
Yeah.
Intervene on
I'm going to try to paraphrase because you were on mic here, but what Felix is asking is about, I think, longitudinal monitoring of the methylation following interventions like yoga or stress management, and you've been able to see the reduction in methylation after those interventions. Did I get it right? Okay.
Yes, definitely. So there are ongoing studies. Actually, we currently have a pilot study looking at changes over time. And one of the key things that we are looking at is meditation and also acceptance commitment therapy. And what we're trying to do is, can we basically reverse it and see when it's going to happen? The current longitudinal studies have been very short-lived, like 12 weeks of yoga. But it would be good to see, which is really what we hope to get to, is see how the changes occur over a longer period of time. So I agree with that comment that there is a need to look at it over time, but also a need to see whether they can stay kind of unmethylated, if that is the best way to describe it, over a longer period of time, post-intervention. So yes, there are a few studies looking at interventions over time longitudinally currently, and they are showing some great promise.
I thought it was interesting when you were showing the pathways, there were other pathways involved, not just the pain. There were cardiac pathways, there are one circadian rhythm pathway. And so you can imagine, I mean, today pain, but I can imagine so many of the other health disparities that we see could potentially be explained through these mechanisms.
Definitely. So one thing that, for me, one thing that has emerged from this work is really the fact that, which is why we highlight pathways as opposed to individual genes, because we have realized that, again, the body doesn't work on turning on one thing at a time, right? So for instance, there is a strong relationship between sleep disorders and pain, right? And when I talk of sleep disorders, it's not just insomnia, but also things like even sleep apnea. All of those have been associated with worse pain outcomes. And we also know that some of those sleep disorders, for instance, have cardiac relationships. So that shows you how a lot of the disparities that we see and most of the chronic conditions have these interrelated or share common pathways. So I have one of my colleagues who does work in cognition, and we are currently discussing how can we collaborate to look at some of the commonalities, not just in pain and cognition, but also pain and other neural processes. So I think for me, what it's really showing is that we cannot continue to study things just in silo, but really collaborate so that we have a broader understanding. Because for a neurologist, if I show you that data, you probably see something totally different from what I would see. So I totally agree.
Yeah. Other question? Oh, okay. So Sneha is asking, in the Parkinson's world, we are seeing more emphasis on genetic testing and the interplay between genetics and environment as causative or prognostic factors to explain the wide variety of phenotypes. What would be your recommendations for looking at epigenetic changes in heterogeneous conditions like Parkinson's? What's a way to break this complexity to something man look at?
Definitely, I think something like there is a room for or space for genetic testing, right? So there are some diseases, Parkinson's being one of those that we know have a strong relationship in terms of the genome, right? But where I always hinge on going towards epigenetics is really for polygenic diseases, right? So if you have diseases which tend to be polygenic and diseases that we know environmental factors, again, luckily, Parkinson's being one of those, if there is a disease that is modified by environmental exposure, such as increased exercise or a healthy diet, right? Then it's telling you that maybe the epigenome is really playing a stronger role than the genome. So would I recommend looking at epigenetic changes in something like Parkinson's disease? Absolutely. I think there is a limitation in looking at gene-environment interactions from using statistical analysis, which is what historically people have done, where you look at the genetic change, they look at the environment and look at gene-environment interactions. The good thing about looking at epigenetics is basically saying, well, let's not look at the environment and the genes separately and see interaction statistically. Let's actually look at what is happening in the body in terms of regulating the genes, in terms of the gene expression in response to the environment. So I think it would be a good way to go. The key is always phenotyping, right? The key for most, be it genetic or epigenetic studies, is always phenotyping. Do you phenotype it enough or well that you can see differences?
Do you have any tips for us on talking to patients about issues? Because not only particularly complicated to explain epigenetics, but trying to convince people that there are actual biological changes that happen from meditation and yoga. Because sometimes patients, when you go through these ideas, will give you a look like, yeah, no, that'll never work. And they don't believe you. And so I don't know, I actually don't know how much you patients in a clinic setting. But for those of us who need to have these conversations, do you have any advice?
Definitely. I mean, I still see patients. Actually, the only difference is I see patients accurately. After this, I'm going to the OR today, actually. So what I have found to help the most with patients is, one, listening to them first and really getting their buy-in for them to see that you care, right? Everything from me stems from the patient believing that as a provider, you actually care about them as a person. Once you have that, it's much easier to introduce some of these complex things with them to say, hey, I know you might not believe it or know you might not have the buy-in, but it's worth the try. This is really where we see things going. It's usually a turn off only when we approach the patients and say, hey, do X, Y, and Z, when they do not even trust us. So developing a rapport with the patient and trust with the patient is really, I think, the critical step in getting towards some of these more advanced interventions. So once the patient trusts you and know that you have their best interest, they will probably follow your lead.
Yes. Rich is saying that yoga is not an option for him personally. Okay. So this question comes from Dr. O'Brien. He's wondering about the spark protein, and did you look at it specifically for a reason, or was it just one of many and it came up?
It is one of the many that, so the goal is always to map from not just to say we see changes in DNA methylation, but to say, do those changes lead to gene expression? So it's one of those that actually map through from changes in DNA methylation to changes in gene expression. Because again, when you're looking at mechanisms, it's not just a single spot. You want to be able to say, yes, this epigenetic change actually leads to a change in gene expression. So that's the one that actually mapped through. And in his comment about yoga, I think yoga is just one of the many interventions. Some people will do mindfulness, some people do acceptance commitment therapy. There are so many other non-pharmacological interventions. So
he's wondering so about Spark. Is it particularly famous, you're asking, for being epigenetically modified?
No, it's actually not. It was actually a surprise when it came out when I spoke with the team that did that study. Basically, they were not expecting Spark to be the gene that came out because most of the hypotheses hinge on things like the mu opioid receptor genes which were already regulating, either cytokines, mu opioids, endocannabinoids, but Spark, which regulates collagen, right, is what came up in that study.
He's commenting that it said regulated in the CSF in older ages and Alzheimer's disease.
Okay. Wow, I didn't know that. Thanks.
Well, I want to say thank you because this was fantastic. I really how we're talking mechanistically about things that we've known were true about the deleterious effects of racism on health. But now we're starting to build explanations that go beyond the surface level saying racism causes health disparities. We've said that for a while, but now we're getting into why and how, and I think it's fascinating. And so I really appreciate you spending the time with us today, and thank you for coming.
All right. Thank you so much.